I'll be talking today about mixture of the basic research and application, hopefully to the clinic. And our interest, as Lady nicely mentioned, is liver disease, viral hepatitis B and C, and discoveries which we made recently in recent years about mechanisms of integration of hepatitis B virus to human genome, and also in the very useful model of hepatitis B, which probably you maybe not heard about, is so-called woodchuck model of Hep B, hepatitis B infection, which is also excellent model of human hepatocellular carcinoma. Yes, sir. Okay. Okay. So just to a little bit information about hepatocellular carcinoma, just that you a little bit was aware, and I'll be going step by step with several information before I start talking really about kinetics of integration. Before that, I say to you about cancer, I say to you about virus, I say to you a little bit about model, animal model. After this, I say to you how we found integration and when they happen in the human genome and in the woodchuck genome. And later I say to you how this happened time from infection to in the first 3 days. So the liver cancer is really much more common that we know from the point of the public. In the clinic, we have, this is the most common cancer, and there is one of the most deadly cancers, because survival after liver cancer is 5 years, is very rare, is less than 5%. Majority of people unfortunately die in the first 5 years, even when they have liver resections, tumor resections, and because the liver transplantation and liver resection, unfortunately, are the most common ways to treat this cancer. And another is much more common in men, twice more than in women, and majority of the hepatocellular carcinoma is related to the neoplastic proliferation of hepatocytes. This is the main liver cells, parenchymal liver cells. Minority is to the so-called cholangiocarcinoma, which is related to epithelial cells, and I'll be not talking about this cancer. So the main reason etiological for development of the cancer is chronic hepatitis B. This is again, and I show you some very briefly data, is really very common infection. It's much more common than HIV. It's common like malaria. So about 1 million cases a year. And the development always involve integration of the viral DNA, because this is DNA virus, although up to recently we didn't know exactly how this happens. The scientists were able to detect integration in the cancer. This looks like terrible, and but we didn't know how this happens, what is the reason and mechanism. So reverse. So just to make you aware, there is at least, and recent statistics as really, because this is like from 2015. From this year, we have really 296 million people which have a symptomatic chronic infection. These people are really sick for life. And although we have therapies, they really do not stop virus replication and the virus progress. in addition, we have 2 billion of people, 1/3 of the human population, which has markers of the asymptomatic infection. And my lab was one of we discovered that many years ago. As I wrote here, this is much more infectious virus. 5 copies of the virus is able to induce infection which persists for life. So the vaccination against Hep B is extremely important. Even single contact with this virus can really change life of the person. So the occurrence is mainly in the Asian and African countries, although we have also quite high occurrence. In some regions, for example, in Canada, we have up to 10% in the north part where infection in the past was quite common, and now we have quite a lot of liver cancer. Hep B, some of you know this well, as we were talking a little bit, that is a DNA virus, partially double-stranded DNA virus. This is important because predisposition to integration is double-stranded DNA breakages, double-stranded DNA. But this is very unusual virus because although its DNA replicates via or through RNA intermediates. So DNA, RNA, DNA in the genome of the virus during replication. This is important because it's prone to mutations like influenza, have a high rate of mutations. And we have we have 6 genotypes, but we have tons of variants, which can be different in each patient, slightly different. There's also a big problem because it is forming mini chromosomes. This double-stranded DNA can be closed, and for this reason is called covalently closed DNA, and this form is very resilient to therapy. The most commonly used therapy right now are nucleoside analogs, small synthetic sequences which can attach to the enzymes important for virus replication in the cells and partially block replication, but not completely, never completely. As I mentioned, the most common feature always is the integration of the virus to DNA. Always this happens. So if this integrate, this is until the cell will die or patient. And this is only like 30 years ago when models, doesn't mean animals, which naturally have a virus similar but not identical to HBV, hepatitis B virus, were discovered. One of these models, maybe you know, this is so-called groundhog. This is in North America, this is in Europe, this is in China. These animals occurring in the mountains. And the virus is very similar, like 70% of the genome is identical. And this have a chronic hepatitis, have a transmission from mother to baby, because the problem of Hep B and other is transmission from mother infected to the baby. And now this is the most common mechanism, not because of the of the blood transfusion like in the past, or intravenous drugs are also quite common, but the mother-to-child transmission is the most. Just to let you know some information, maybe you'll be useful. So vaccination again is extremely important. So we can start also mother-to-child transmission in this model. Okay, so what was interest in? I studied this virus from my medical school. Okay, so I am, as you can see, quite advanced in my age, but not so much, just looking so this way, but inside I'm very fresh. The point is this, that we studied this virus, I am studying this virus for a long time, but in the recent years, I was always asking the question, okay, a virus integrates, but when this happens, what is the time frame? Because in the past, in the clinic, because I am also MD, we're thinking that the integration occur approximately 2 years after infection, based on the very low sensitivity techniques of study integrations. But anyway, this was our recent questions in the last maybe like no more than 7 years. And another question was, what are the sites of integration in the human? What is the sites of integration from the point of the virus, where this happens? And how fast this happens? What are the kinetics and what are the mechanisms? Anyway, I try to say to you this in the next part of my talk. Okay, but not in this way, only reverse. So one of the first studies which we did, and it was already a few years, like 5 years ago, published and done 7 years ago exactly, we were interested, recognize what are the initial sites of integration in human hepatocytes in the culture and in the woodchuck model of Hep B. So we studied, we take the virus, infectious from patient, not construct, not, and this is important what I try to say to you, virus from constructs, which we do now commonly, not always behave the same way as a naturally occurring virus. And this is quite important because we see this in Hep C, we see this in Hep B. Yes, you discover something in the synthetic virus, but you need to really prove that this is in the nature. So we start the infection after, as you can see, from 15 minutes to 7 weeks in this first study in these cells, which are good cells. We know that they infect, that the virus can infect them. And we also study in the woodchucks. So we infect the animal again with the wild type virus, not any kind of the constructs. And I took liver biopsy before experiment, and after infection 1 hour, inject the virus intravenously. And after 1 hour, I am taking liver biopsy, and in another animals after 3 hours, and here was 6 weeks, 2 as a control, because in 6 weeks, we usually have an acute hepatitis, acute symptomatic infection. We used a technique which was not developed by us, but developed by other people, but we applied. And the postdoc who was doing this was really very skillful and excellent. This is precision, this is tough work, and he spent hours and many other my students on this to establish this technique. Anyway, this is so-called inverse PCR. We're using primers to prime the virus, but we are extending replication, amplification, because this is based on the PCR, to the host sequences which are flanking the virus. So we discovering the sequences, we can now sequence them, and we can go to the gene bank or to other bank and see what they are host sequences associated with the virus. Anyway, everything is published. So we were really fortunately successful. We were able to, on the single nucleotide level, dissect where is the virus, where is the host sequence, and this is gene which we discover integrating in very fast in 1 hour after the infection. After exposure to the virus, I'm saying exposure to the virus, because infection and exposure, this is not the same thing. And we found, for example, here, and this is in this human line, hepatocyte line, and we found also that this integration to line 1, this is retrotransposon, and I try to say to you that there are 50% of the human genome are non-transcribed sequences. And the big group of that are also transposons, retrotransposon sequences. And I will be briefly later mention about their significance. But anyway, here we have a very beautiful recognition of the sequence without no problem. There is many other problems, but in these sequences, we have no issues to know which part of the virus integrates to which human gene. And this was repeated in several clones, as you can see here, 8 clones, and this is 6 clones. So this is very strong indication that this is indeed true. And we were able to identify the place in the chromosome where this happens. So we have really full stuff, full recognition. And we found that very early in this work, in this study, we found that even in 1 hour, we can detect virus integration. I have to say to you, because from other study, which I don't have a slide for here, in this we found that in 1 hour, we also see virus replication, appearance of the RNA, this RNA, which I mentioned, this transcripts RNA, and DNA, of course, yes. Anyway, this is what we published. You can see this. This is good paper in the Nature Oncogenesis. You can see that we are detecting at the beginning of the infection this very early, between like 1 hour, 24 hours, we detected few different kinds of the genes, which majority we can identify in the human, but also we detect retrotransposon sequences. And I say to you later, this study was confirmed by other group from Germany that they found similar kind of 1 year, published what, I think 1 year later, that they found similar, that transposon and retrotransposon are quite common sites of this virus integration. In the woodchuck, we have found very important point was that yes, even in 1 hour in the natural model of infection, not in the culture, we are seeing the same thing. We can detect and we mapping them, we done where this chromosome are located and what this happens. And I will be not saying too much. This is, we identify the site of the break, the virus DNA, which is associated with the human genome, okay? So we found the particular regions are the most common sites of integration, which is so-called X, and in the X gene, some particular regions. I will be not reading this. This may be one thing I mentioned, that in the study we found that up to 45% of integration sites are in the transposons and retrotransposons. Why? Because next study we focus on that. And this is study done with a collaboration with our colleagues from the Tokyo National Institute of Infectious Disease, excellent people, which established a very good model in vitro because they transfected the human-like cells with the receptor for the virus, which was recently discovered, like 10 years ago, because for 40 years we didn't know what is a receptor. It was very tough. But so this is a very efficient system. We use the system because we really want to study retrotransposons and this, but also really whether in this very efficient system, whether it's improvement in at the time of integration. Yes, we found that we can detect integration in the 30 minutes even after injection of the virus or administration of the virus. Again, we found indeed that there is a lot of retrotransposon sequences. I'll be not saying to you, this is like small discipline now. It is talking about meaning of the sequence. And again, we have a nice recognition. This why I'm paying attention to this retrotransposon? Because these are sequences which can integrate, for example, with the viral sequences, like in our case, and transmit the virus sequence to the other chromosome, to another gene, to another location, without infection. So it's spreading, and this virus, our virus, is highly pro-oncogenic, because I didn't maybe forgot to mention you that the cancer, if you consider the death from this cancer, is third in the world, is very oncogenic virus, much more oncogenic than the tobacco overall, if you take cases. Anyway, this retrotransposon played many different roles, and we are discovering more and more in evolution, in how we change our genome, but also looks like that the virus is using this natural process probably to spread through the human genome or animal genome to establish often integrations across chromosomes. And this we found the most common place is line 1 retrotransposon, and this particularly was found with a highly endogenous mutant, which is really known to able to relocate genetic elements to other places. And it is it was proved that this HBV line 1 integrations are really can spread through the genome, and they are really can be oncogenic in vitro conditions by other groups. So it is connection with the pathogenesis of the HCC. It means hepatocellular carcinoma, means oncogenesis in the HBV-infected hepatocytes. So now when we did that, I still have a question: what is going on, and really, what is the mechanism of these integrations, and exactly what are the potential stages of this? So we use another cell line which we established from the woodchucks, from the healthy woodchucks. And we, again, done infection experiment, this time even like in the first from 15 minutes up to 72 hours, means 3 days. We didn't went further. We know what happens later. We know that the number of integration increases during even these 3 weeks, which we studied. So we know that. I want to mention, which again, sorry that I'm jumping a little bit, that this integration, which I show you in the second study, all of them have the same format, so-called head-to-tail junction. And in this study, we discover first, which was further surprise, that we can detect in 15 minutes. So even more efficient system, woodchuck, than the human cell lines. And that all what we detected is, this is important because this head-to-tail junction is because of the particular molecular mechanisms. So by this, we already have by work of other people, we have a clue what we need to investigate. Of course, this does not mean that there is no other junctions. This, our calculation is approximately 95% junction discovered, these initial junctions are this head-to-tail junctions. So you can see just like that, head-to-tail. But about 5% is overlapping. This one strand, another strand like that. Okay, so it's a so-called mini homology. And this is different mechanism. People will recognize what is going on and how this can be formed and what are the differences. And also, I just want to say to you that yeah, we were somewhat smart, but not. We used what other people were investigating, and I summarize this briefly. This is what I said almost, but this we know before, that the hepatovirus is, of course, DNA, double-stranded DNA virus, that the double strand, and these are really precursor for integration because the double strands in HBV breaks. So, and we also know from studies, other people from our research field that HBV can induce oxidative stress. And oxidative stress is one of the most common, but not only, mechanism of the breaking of cellular DNA. And that this, we know also that these breaks could be or are the substrate for creation of virus-host junctions. So these are like, just from this, what I learned during the meetings or from papers and so on and so on. So it was like, for us, was like obvious what to do. So we, how to solve the issue, what happens, how this initial junctions are formed. So we need to determine if and when HBV and woodchuck virus induce hepatocyte DNA damage, whether the virus induce very early oxidative stress inside the cells, and about if we know that, there is also clues about the mechanisms of formation of integration. And from our study, we learned that the majority of the non-homologous joining is this head-to-tail junctions, and we know that this is induced, and this is like biochemistry, okay, in this moment, like mechanism which is quite well recognized at this moment, so-called PARP1 dependent DNA repair machinery. And I show you briefly how this looks like, that you can see what it's about. But anyway, so okay, we did the studies, and now as a first part, we ask this question: what is really happening? It is, if we start doing in 50 minutes, what is going on from the point of the virus DNA presence inside the cells, RNA presence means replication, and integration. So integration we found right away, but replication later, like you can see in 1 hour. Of course, it is not so much maybe difference between 50 minutes and 1 hour, but, and these are very sensitive techniques, all of them similar sensitivities. But anyway, this suggested that integration occurs before replication, which is itself a little bit surprising. So the next step, we study whether virus can damage DNA. And these are classical assays, like probably many graduate students know. This is the alpine DNA comet assay, which you put under fluorescent microscope and you study the length of the tail on this, for example, here. This is now, you have a microscope, you have a computer is doing that. So it is, creating you statistics we study in the 50 minutes to 1 hour, and you can see that comparing to control in the 15 minutes, we have a significant damage of DNA. And in 1 hour is really big one. So yes, virus in 15 minutes already is inducing DNA damage of in hepatocyte. And now, okay, whether the virus is inducing oxidative stress. And my postdoc Ranjit, I'm repeating, it's really, it established assays for all of this, what I will be showing. PCR assays or expression or induction on enzymatic assays. Anyway, point is this, that ROS is reactive oxygen species. So like H2O2, similar to that. So we know that they are really highly destructive, doesn't mean they're breaking DNA. Another is INOS, which is nitrogen species, which is also can do similar thing. And as you can see, in 15 minutes, we have a really huge jump in the oxidative of the presence of oxygen radicals after exposure to the virus. And this persists for some time, but the really peak, as you can see, of course, it's later, it's non-specific because background is starting increasing. It's really, the star means that this is the peak. INOS, no. INOS was only at the beginning, in the first 30 minutes, but after this was not so involved. So this, of course, indicates that this is like main thing induced by the virus in the destruction, in the breaking of the DNA in the hepatocytes. And of course, here is some control, so maybe not control, but another kind. It is really expression of the, and this is the PCR technique, so this is not enzyme activity. But anyway, it's activated also this hem oxidase 1, which is a response to the oxygenative stress. So it's a confirmation that indeed oxygenative stress was taking place in this cells. Okay, so we have a DNA damage, we have an oxidative stress, oxygenative stress, so we know that at least these two elements are working here. And now, because of this, because PRP mediated mechanism to a large degree, but not only, but to the significant degrees induced by the oxidative stress, we focus on that. And we're not saying that only this could be involved, but anyway, I know it's a very biochemical, very, progress is huge in this area in the last, I don't know. I'm not like expert in this particular machinery. But anyway, we analyze this machinery and we design the enzyme and the methods, assays to detect the particular elements. For example, this is oxidative damage. So the one of the first is this OGG1, which is enzyme which is removing guanidine, oxidative guanidine, which is oxidated during the DNA damage as a first step. And after we start the expression of the PARP1, and after we start the expression of this protein, which is like very tightly associated with the PRP1 to confirm that this is PRP1 and so on, just to have a few elements to recognize whether we are really doing and detecting this what we supposed to detect, or we think that we should detect. Anyway, as you can see, this PRP was activated very soon after infection, after exposure to the virus very early, as well as other elements of the PRP repair, DNA repair machinery after exposure to the virus. And here, this was supplemented by two other factors, by two other elements, which is, this is again quite common, enzyme, which is in response to the DNA repair machinery, but not only, in general to the cell stress, but when this is PRP mechanism, this is always an ADP plus increase, expression increase, and the cleavage of the PRP1 after termination of this process. So this was increased later, like you can see, like from 6 hours, but also the significant differences was just from 15 minutes. So the degradation of the PRP1. Anyway, we think that we have a good indication what happens. And this is like summary, whatever, slide, or which we show these different markers, which we see the integration from 15 minutes in the system, which we have a very evident oxidative stress, less nitric oxide stress, and we have all elements. We have a DNA damage, which you can show by the simple fluorescent assay, and we have all elements, or majority of the elements, confirming each other that this is activation of the PRP repair machinery. So this is what I want, I think, to say. And I have a couple of more minutes, not so much, but so that this initial urease integrations are really dependent from the double-stranded DNA damage and initiation of repair. So the virus is using naturally occurring process, physiological process, to integrate to the virus genome, to the host genome, to the cell genome, because this is the regular, normal repair mechanism after oxidative stress and other injuries. That this predominant tail, head-to-tail, confirm that this is PRP1 mediated process, because this process is really, how to say, mediate the non-homologous enjoyments. And yeah, this is like, we're thinking that the same is, although we didn't done side by side HBV, because HBV experiment in this particular study, we are thinking that the same applies to the human HBV infection, because of the really very significant compatibility between because these viruses and the processes of infection, replication, and pathogenesis of liver disease and cancer. This person, Ranjit Chauhan, was like he did a lot, or majority, and I have other people contributing to the studies, and have a very nice collaborators from Japan, and I'm quite well supported by Canadian organization. And this is our, just to let you know a little bit from the place which I am. So this is like Puget Island, close to, on the east coast of Canada. Not too many people, 250,000, on the size of France. So it is not a lot, but we have a beautiful university. We have a lot of students, graduate students. This is hospital, and this is a university which is far away up to there a little bit. And we have unusual things, which is, if you will be there or close by, it is worth it to see. We have maybe Newfoundland dogs and whales and icebergs and Vikings and the big fish as my student catch. Thank you very much. Thanks.